Hello. In this video, we will look at the coronavirus and its genetic information. Today, the WHO declared a worldwide pandemic of coronavirus, and the whole world is concerned about it. So we thought we could take a look at the more detailed information about the virus and to understand it better. This virus started appearing among humans near Wuhan in China, and it's now called as SARS-CoV-2 or COVID-19. For the full form is severe acute respiratory syndrome coronavirus 2, and this particular page in the NCBI NIH gene bank tells us the detail about this virus, and it gives the genetic code, as you can see here at the bottom. It's about 30,000 letters long. And so what we're going to do is take this information from this page and try to see what we can understand about this virus and how we can use our platform in Z to make sense out of it. So now that I have dragged and dropped this information into the content or Z space of ZCubes, let's see if we can take a look at this information that we are seeing here. And the first thing we'd like to do is to extract out the genetic information, which is basically like ATT, AA, AG, TT, and so forth until the end. That's it ends in a series of A's. And that's kind of typical of these types of viruses, that it ends in such a sequence of repeated A's. As you can see, the text closes with 2 forward slashes at the end, and the start of the genetic code starts with origin here. So that gives us an idea of how to extract information from this cube. And you can see that this cube is named as 2 space, as the tooltip says, or when you take the mouse and enter this cube, you can see at the bottom here that it says 2 space, and that basically means that this cube is called 2 space. So to extract out the information from this cube, let's take this approach. The first thing we'll do is to say ztext2 will give you the text from the cube number 2, that is 2 space, and we will split this data, the text that we get from here. So if I did ztext2, it'll give you the content from that cube. So let's take a small piece of code and call this as, with this small piece of code, where we take the text from cube 2 and we split with a regular expression starting origin, and we split this entire content text with origin and take the, so you'll have 2 pieces, and you take the right side of it, that's basically the index 1, and then you split that piece to find the 2 forward slash, and so you'll have again 2 pieces. You take the first piece of it, and then you replace, as you can see, there are a lot of line numbers and stuff, and if you could just remove the numbers from here and clean up the space, you basically would get the genetic code, and that's what we'll be doing. So the first thing we do is, there you go, there you have the genetic code, and we assign that to the variable called COVID code. Now that basically means that we have the information all in one place, and now we need to split this into pieces of 3, like ATT, AAA, GGT, and so forth, to extract out the mapping to the appropriate amino acids that will eventually form the proteins. And you can see that in this listing here, it's indicated that the coding sequence from 28274 to 9533 is basically coding for this particular protein, and this genetic code corresponding to that will convert to this translated code. So let's see if that's kind of the case. So to do this, first thing you need is the mapping between the amino acids and proteins, the genetic code, the amino acids that form the proteins. So here we have a page which you can just search for codons and mapping to proteins, and it'll kind of come up. And here in this table, it has the mapping between a particular genetic code and its amino acid equivalent. And so what we'll do is we'll take this text, we'll select this text here, and then drag and drop this into ZSpace. So now you have the codon protein mapping here. Now that you have it, we will extract out the codons from here. So now we have the table, and the table ID is table 1. As you can see in the tooltip, it says table 1, and also it shows at the bottom left here when you take the mouse over it that this table is called table 1. So to access the information inside the table, we could simply say pound table 1 and it'll get the information from that table. So if I select this and run just that, it'll show you that the content of the table is now available to you as an array. And let's name it as codon mapping. So now codon mapping has the information of the table. Now, if you wanted to look up a particular codon, like let's say TGA, just like in a spreadsheet, you would say we look up TGA in this array codon mapping and bring the third column and tell me what it is. So it says that TGA it maps to the amino acid TER. So if you had TGA, if you could go down here, let's see where TGA is. There is TGA, it's a termination, or the amino acid code is X. So that's how you could look up from this table and to get any particular mapping between a codon and a amino acid. But this could be fairly slow if you started looking up for every codon in this particular sequence. So let's take a different approach. Let's just create a lookup table. So we will call this as CLOOKUP. It's simply an object. And we go through the codon mapping table that we just created. And we will go row by row. And for each row, we will take the first element in that row. So let's say TTT, and we will assign CLOOKUP TTT to be this row. And the next one is TTC, so it will say CLOOKUP TTC is equal to the second row, and so forth. So now this gives you a very quick lookup by the first column code, and now CLOOKUP can be used for doing lookups on the codons. So let's go back to the codons now. So the COVID code is basically the genetic sequence that we extracted here. And what we are doing is we are splitting it first and taking chunks of 3. So this will basically give you a set of, like ATT, AA, GGT, and so forth, 3 at a time. And then we convert them to uppercase so that it can now be mapped to the CLOOKUP object we just made. So wherever the CLOOKUP exists, it's going to take the third column from the CLOOKUP, which is going to be the amino acid code. And if it can't find it, it'll just attach UNK. And eventually it'll be, all of these will be converted to a string. So now what's happening here is that it has taken the entire code of the coronavirus genome and given us the potential amino acid sequence if the entire RNA was converted to a humongous protein. But that's not what that happens. Obviously, as we can see in the information here, it is taking different genes in the sequence and it generates the amino acid sequence corresponding to that. So let's get into the next level, in which we will take this same logic that we created here and we will make it into a function. So we will create a function called amino acid sequence, which basically, given a sequence, will split it into chunks of 3, uppercase it, and then it'll look up from the CLOOKUP object, and then if it doesn't find a match, it'll just add an empty string, and this will give you the amino acid sequence function. Now, as you can see in this sequence that we have here, it says in this particular case, in the genetic code from the 21,563 third location to the 25,300 that much is a sequence that codes for the structural protein. So let's do that. And here we're going to use one more feature of ZCubes, which is that you can actually use long variable names. So you don't have to say structural protein as one word. You can use it as 2 separate words with a hash to prepend it. So you can use variable names with spaces. So structural protein variable name now will be filled with the amino acid sequence by slicing these 2 numbers. Like as we saw here, it was saying that the structural protein is from 21,563 to 25,384. So that's what we are using here. And we subtract 1 because our index is starting from 0. So if you run this, you get this translation of that genetic code to its corresponding amino acid. So it starts with MFV and it goes on and goes all the way and it ends in HYT. So let's see if that is correct. So if you go here, it says it starts with MFV and it ends with HYT. So this translation is correct. So now we have in our structural protein sequence, we have found the appropriate coding. So similarly, we can make all the other protein sequences that we have in this on the left that you see here. All of these genes, we can just try to search in this sequence and generate them. So that's what we are doing here. So if we took the ORF3 protein, it starts from 25,393 to 26,220. And if you look for it, it will be MDL and so forth. So if you go for ORF6 glycoprotein here, ORF3A protein from 25,393 to 26,220, it starts with MDL and it goes on and ends with VPL. So if you were to come back and look at ORF3 protein in the sequence that was given in the gene bank, ORF3A protein starts with MDL and ends with VPL. Yes, that's exactly what we have found. Now there is, if you went through all these sequences that we have generated here, they would match exactly as the gene bank says. ORF10 glycoprotein starts with MGY and ends with NLT. So as you can see here, it starts with MGY and ends with NLT. Now, if you look at the very first coding sequence, the ORF1AB polyprotein, this is a little bit more complicated than the rest of them because it's actually a segment from 266 to 21555. But if you did that, you will not get this exact sequence. The first part will be the same, but the second part, after some time, it'll not match. And that's because there is a frame shift that happens. It's called a ribosomal slippage that happens at the 13,468th character or that code, at which it it slips and goes back a little bit. So what we have done is we have figured out two separate protein sequences. So the first one is this one. So it starts with MESL and it goes till it's a long sequence. So QSFL. So this is the first part, MESL. So it's MESL here, and it ends with QSFL. So let's see where that is. QSFL. So it ends here, and then it's supposed to start with NRVCG and so forth. However, if you did do this, it doesn't start with NRV, it starts with RV. So N is missing. So what happens is at this point, around this point, so we have done a little bit of research on this. So if you take around this area from 13450 to 13500, this is how the code looks. Now if you started exactly from 13468, it starts with CGGGTTT. So let's come back here to the coding around it. CGGGTTT is right here. So our missing N must have happened by a small shift around this area. So let's find out from the codon mapping what is the mapping for getting an N. And as you can see, in this list, there are two sequences, AAT and AAC, that could have given us an N. So you would see that. So if you went and looked at this table, AAT or AAC could have given us an N. And what happens here is AAT or AAC could have given it. So basically, it kind of skipped back a little bit and read from AAC onwards and this AAC onwards, and then it went back one and started with CGG. So that's how the coding actually went. So the very first one is a little bit, this particular protein, the OLF1AB polyprotein, is a little bit tricky in that it was not a straight read. It kind of paused in between, bent back a little bit and started reading it again. So effectively right now we have explained all of these different pieces of code. So if you take structural protein, starts with 26523. So if you look at structural protein, starts at 21563. Let's take an example of this particular one, membrane glycoprotein, which is a structural protein, but membrane glycoprotein starting at 26523. So if you take a look at membrane glycoprotein starting at 26523, the code starts with MAD and so forth, and it keeps on going like that. So and that's what you see here. So all of these can hence be generated from this code in a very quick fashion, given, like, if you know what those indexes are, using the codon mapping, we can generate the appropriate amino acid translations. And we have basically applied this technique and gotten all of these genes explained from the information that we have now. So that's an example of how we can use the capabilities of the system to take data from different data sources and analyze and get results that can now be used for further analysis. Because now, for example, this particular variable has that value, you can go and write some code on that and try to do a further analysis, maybe pattern matching or checking with some other protein or so, or maybe even you're doing drug discovery, where you can look at how could you probably break pathway in this protein or so. Many different uses you can do with this tactic. Any comments, please leave a comment below, and thanks for listening.